So let's look at some of the basic features of, or the different features of antimicrobial drugs. We can class antimicrobial drugs as antibacterial antibiotics, antifungal medicines, antihelminthic medicines, antiprotozoan medicines, or antiviral medicines. Sometimes with antihelminthic and antiprotozoan, we call them antiparasitic as well, drugs. But these are going to treat those specific microorganisms, or some of them could be potentially macroorganisms. So how do these drugs work? They're either going to inhibit microbial growth. Any drug that is called a static drug is going to inhibit growth, whereas a cytal drug kills the microbes outright. And we saw this back in chapter, I think, 5, when we were looking at, it might have been chapter 4, I think it was chapter 4. We were talking about the different chemicals. We can actually determine the amount of the chemical that we need to inhibit the growth and the amount of chemical that we need to kill off the microbes. So the MIC, or MIC number, is the lowest concentration of an antimicrobial agent that inhibits the growth, whereas the MLC number is the lowest concentration of antimicrobial that will kill off the microbe. Some antimicrobials are used in combination with each other. This is typically seen when you have an infection that is hard to treat, and one of the drugs may be has a synergistic effect on the other drug, and then when one drug functions, or when one drug kills off the infection, the other drug can inhibit the growth of other microbes that might come in and take the place of that infective agent. So there are some tests that scientists use to determine the level of antimicrobial activity and what antimicrobial might be best used to treat different infections. The most famous one is the Kirby and Bauer disc diffusion test. This one we use a special type of agar, and then we have the minimal inhibitory concentration test as well. So with the Kirby and Bauer disc diffusion test, this is a standardized test that is used to determine which chemicals are going to be best used to inhibit or to kill off growth, and which antibiotics are best used to kill off microbes. We use a Mueller and Hinton agar that we produce. We then plate different microbes, whatever we're concerned about, be it Staph aureus or E. coli, and then we take discs that contain certain drugs and place them onto the agar. You can also use sterile discs that you dip into certain chemicals. So you can test basic household chemicals. You can test specific antibiotics. You then grow the microbes under normal environmental conditions, so like at 37 degrees Celsius for 24 hours. And then you look at what zones are found around each of the discs. So here's an example, and there's a very little zone around this one, and you can see that small zone around here. But then you can see a much larger zone here and a larger zone here. So these antimicrobials are going to be better at inhibiting the growth of this microbe. These ones were not very effective at inhibiting the growth. The larger the zone of inhibition, the better the chemical works. So how do these chemicals work? We're going to look at different modes of action, including or antimicrobials work with using different modes of action. One of those modes might be to inhibit cell wall synthesis. So any of your beta-lactam drugs, these are things like penicillin and amoxicillin, they work to inhibit the cell wall synthesis. They inhibit peptidoglycan. Some drugs work to inhibit protein synthesis. Again, without producing the proper proteins, you can't divide. You can have drugs that inhibit nucleic acid synthesis. So without the DNA production, you can't replicate the DNA and then you can't divide. Or you can inhibit metabolic pathways so that the cells can't produce energy like they're supposed to. Last thing we're going to focus on in chapter 20 are these new super resistant bugs. In the 1930s, mainly, well, I guess I should say in the 1940s, when penicillin became available to be used, penicillin was given out like someone might give out candy at a corner store. It was super popular and everybody was using it. But just like any organism, if environmental pressures occur, then organisms are going to change. They're going to adapt to those environmental pressures. And so penicillin was seen to kill off staph very effectively. Staph adapted. And now we have these methicillin-resistant staph. We have these penicillin-resistant staph. We have vancomycin-resistant staph. So really what we have is a multiple resistant Staph aureus. And that's just one example. Resistant bugs are found everywhere. And I thought this was a really cute cartoon picture of a superbug chasing an antibiotic. And I have a video. This is a TED Talk about drug-resistant superbugs. If it comes up. If it doesn't come up, then I'll just tell you that you should go and check this video out. But it looks like it might come up. There it is.
What would you do if your hips and knees didn't hurt? Would you start doing the things you love again, like traveling or fishing, or surgery and physical therapy?
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total body mass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table.
What would you do if your hips and knees didn't hurt? Would you start doing the things you love again, like traveling or fishing? From surgery to physical therapy.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria like Salmonella even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strands that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolines by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria. And although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments.
So what can we do?
Other scientists are investigating alternate solutions such as phage therapy or using vaccines
Make sure
to prevent
that
infections.
we are taking
Most importantly,
drugs
curbing the
that are
excessive and
appropriate
unnecessary
for
use
what
of
we're supposed to be
antibiotics, such
killing
as for
off.
minor
Oh,
infections
hold
that
on
can
a
resolve on
second.
their own,
Sorry.
as well as changing
Oopsie.
medical practice to prevent hospital.
All right. So last thing we're going to just cover is what we can do. Anytime you have an infection, you want to make sure that you're taking the proper drugs in the appropriate amounts. So follow what your doctor tells you to do. Sometimes you may have to take 2 or more drugs at the same time to make sure that you're killing off those resistant microbes. Don't use drugs when you don't need them. If you have a viral infection, an antibiotic is not going to work. Viruses are non-living. Possible solutions for the future, we could find new antibiotics, stop using antibiotics, and let our own immune system fight the infections off, producing bacteria that are no longer resistant to these same antibiotics. I just said some of these. Use other treatments other than antibiotics, like using bacteriophages to treat bacterial diseases. So that's the end of this chapter. I'm going to stop this and I will upload all of these videos shortly. Bye.